Welcome to MPharm Masterclass. Today, we're diving into the fascinating world of neurotransmission, the process through which nerve cells communicate with each other to control everything from heart rate to muscle movement. Visual. A neuron diagram showing dendrites, cell body, axon, and synapse. Section 1. General aspects of neurotransmission. Neurotransmission is the fundamental process by which nerve cells, known as neurons, communicate with each other or with other target cells such as muscles and glands. This electrical and chemical signaling is critical for every function our nervous system performs, from controlling our heartbeat to complex thinking. Visual. Close-up of the synapse, showing synaptic cleft and receptors. Neurons are unique because they don't physically connect. Instead, they send signals across a tiny gap called the synapse. But how do these signals actually travel? What happens at the molecular level during neurotransmission? Section 2. Steps involved in neurotransmission. Step 1. Synthesis of neurotransmitters. Visual. Inside a neuron, showing enzymes converting precursors into neurotransmitters and storing them in vesicles. Neurotransmission begins with the synthesis of neurotransmitters. These chemical messengers are produced from precursor molecules, typically amino acids or other small compounds. Specialized enzymes help convert these precursors into neurotransmitters, which are then packaged into tiny sacs called synaptic vesicles within the neuron's axon terminal. Step 2. Release of neurotransmitters. Visual. An action potential traveling down the axon, leading to vesicles fusing with the neuronal membrane. When an electrical signal, known as an action potential, travels down the axon, it reaches the synaptic terminal. This signal triggers calcium channels to open, allowing calcium ions to flow in. The influx of calcium causes the vesicles containing neurotransmitters to fuse with the neuron's membrane, releasing the neurotransmitters into the synaptic cleft, the small gap between neurons. Step 3. Binding to receptors. Visual. Neurotransmitters diffusing across the synaptic cleft and binding to receptors on the postsynaptic neuron. Once released, neurotransmitters diffuse across the synaptic cleft and bind to specific receptors on the surface of the postsynaptic neuron. Depending on the type of neurotransmitter and receptor, this can trigger either an excitatory or inhibitory response in the postsynaptic cell, generating an electrical impulse or inhibiting one. Step 4. Termination of signal. Visual. Enzymatic breakdown of neurotransmitters in the synapse or reuptake into the presynaptic neuron. The final step in neurotransmission is the termination of the signal. This is done in two main ways. 1. Enzymatic degradation. Enzymes break down neurotransmitters in the synaptic cleft. 2. Reuptake. Neurotransmitters are reabsorbed back into the presynaptic neuron through specialized transporter proteins. Both processes ensure that the signal does not persist longer than needed, resetting the system for the next signal. Section 3. Neurohumoral transmission in the autonomic nervous system, UNS. Visual. A split screen showing the sympathetic nervous system and the parasympathetic nervous system. The autonomic nervous system, or UNS, controls the automatic processes in our body that we don't consciously regulate, like our heart rate, digestion, and respiration. It's divided into two branches, the sympathetic and parasympathetic nervous systems, each with opposing effects. Visual. The sympathetic system highlighted as fight or flight, and parasympathetic system as rest and digest. The sympathetic nervous system is responsible for the fight or flight response, preparing the body to respond to stress or danger by increasing heart rate, dilating pupils, and redirecting blood flow to muscles. In contrast, the parasympathetic nervous system activates the rest and digest mode, calming the body down by slowing the heart rate, enhancing digestion, and conserving energy. Visual. Diagram showing the release of adrenaline from adrenal glands in the sympathetic system and acetylcholine from neurons in the parasympathetic system. Two neurotransmitters are at the heart of these systems: adrenaline, also known as epinephrine, in the sympathetic system, and acetylcholine in the parasympathetic system. Let's take a closer look at how each neurotransmitter works and how they help maintain the balance between these two systems. Section 4. Neurotransmitter 1. Adrenaline, epinephrine. Visual. Adrenaline molecules binding to adrenergic receptors on cells, showing heart and muscles activating. Adrenaline, also called epinephrine, is the key neurotransmitter in the sympathetic nervous system, responsible for what we call the fight or flight response. This rapid response prepares the body to react in stressful or dangerous situations. Mechanism of action: visual, stress signals activating the adrenal medulla, releasing adrenaline into the bloodstream. When the body senses stress, the adrenal medulla releases adrenaline into the bloodstream. It binds to adrenergic receptors on target organs, specifically alpha and beta receptors. This interaction triggers a cascade of changes to prepare the body for immediate action. Visual, adrenaline binding to receptors on the heart and lungs, showing increased heart rate and bronchodilation. These physiological changes include: 1. Increased heart rate and cardiac output to pump more oxygenated blood to vital organs and muscles. 2. Dilation of pupils to improve vision in low light. 3. Breakdown of glycogen into glucose, giving muscles a quick energy source. 4. Bronchodilation, or the widening of airways, to enhance oxygen intake. Key functions: visual, summarizing adrenaline's key effects: increased heart rate, blood flow to muscles, bronchodilation. Adrenaline's key functions in the sympathetic nervous system include: 1. Cardiac output. It increases heart rate and the force of contractions. 2. Blood flow. Adrenaline shifts blood flow to muscles and away from non-essential organs like the digestive system. 3. Bronchodilation. It opens up the airways in the lungs to allow for deeper, faster breathing. Termination of action: visual, enzymes breaking down adrenaline in the synaptic cleft and bloodstream. Adrenaline's effects are brief, thanks to enzymes like monoamine oxidase, MAO, and catechol O-methyltransferase, COMT, which rapidly metabolize it. This ensures that the fight or flight response is swift but does not persist longer than necessary. Section 5. Neurotransmitter 2 Acetylcholine H. Visual. Acetylcholine molecules binding to cholinergic receptors on different organs like the heart and digestive system. Acetylcholine is the primary neurotransmitter in the parasympathetic nervous system, which regulates the rest and digest functions, calming the body and promoting energy conservation and healing after stress. Mechanism of action: visual, acetylcholine being released from a neuron and binding to receptors on muscle cells and organs. Acetylcholine is released from the parasympathetic neurons and binds to two types of cholinergic receptors. 1. Nicotinic receptors. Found at the neuromuscular junction, they mediate the contraction of skeletal muscles, allowing voluntary movement. 2. Muscarinic receptors. Located in organs like the heart, lungs, and digestive tract, these receptors regulate involuntary functions such as slowing heart rate and stimulating digestion. Visual. Binding of acetylcholine to muscarinic receptors on the heart, showing a decrease in heart rate and smooth muscle contraction in the intestines. Once acetylcholine binds to these receptors, it triggers specific responses depending on the receptor type and organ involved. Key functions: visual, summary of key effects, decreased heart rate, stimulated digestion, smooth muscle contraction. Acetylcholine's key functions in the parasympathetic system include: 1. Decreased heart rate. By binding to muscarinic receptors in the heart, acetylcholine slows the heartbeat, promoting relaxation. 2. Increased digestion. It stimulates the secretion of digestive enzymes and enhances the movement of food through the digestive tract via peristalsis. 3. Smooth muscle contraction. Acetylcholine causes contraction in smooth muscles, aiding in processes like digestion and bladder control. Termination of action: visual, acetylcholinesterase breaking down acetylcholine in the synaptic cleft. Acetylcholine's action is terminated rapidly by the enzyme acetylcholinesterase, which breaks it down in the synaptic cleft. This ensures that acetylcholine's effects are precisely controlled and do not last longer than needed. Conclusion: visual, summary slide of neurotransmitters and their effects. To sum it up, neurotransmitters like adrenaline and acetylcholine are crucial in regulating our body's responses, whether it's speeding up during stress or slowing down for rest. Understanding neurotransmission is key to appreciating how our nervous system controls everything from heart rate to muscle movement. Thanks for watching M-Pharm Masterclass. Don't forget to like, share, and subscribe for more in-depth pharmacology content.